Okay, that was an absolutely wonderful talk. I'm happy to say and introduce our next introducer. That's definitely not the right way to say it, but um, Dr. Rui Gardner is joining us today along with members of his facility who are very near and dear to my heart, all of them, because I've worked with them in the past, and I'd like to introduce Dr. or Rui Gardner to give us a brief introduction to his lab and subsequent talks from his facility on their activities. So Rui, you are sharing. It looks good here. You just need to unmute yourself and we'll make sure that everything goes okay.
Hello, hello. Can you hear me?
We can hear you. Okay, I will walk away now.
Thank you so much, Kathy, for the introduction. Hello, everyone. I can't see anyone, so I don't even know how many people are there. I apologize for not being there in person. I had a small accident playing soccer, so it will take me a few months to recover and walk again. But I couldn't help but participating in the annual flow cytometry event in New York, which is MetroFlow. So I thought that I at least try introduce the my team, the project. So my idea here is just in 10 minutes kind of give you an idea of the projects we're working on in the facility. Why are we working on projects in the first place? And give you just a few examples, and then Magda, Mark, and Barbara are going to present more details, the projects that they've been involved directly in more detail. So let me just grab here the pointer. Let me see if I can just grab the laser pointer, then it will be easier to point. So most of you guys know where Memorial Sloan Kettering. I mean, this audience is, mostly from New York and New Jersey and surroundings. But just to give you an idea, I mean, it's a very central place, just in the same block. MSKCC is together with Cornell, Rockefeller University, NY Blood Center, and then we have, a bit further away, but still in Manhattan, NYU, Columbia. I mean, there are a lot of places where, collaboration can be thriving. And so it gives us a lot of opportunities to really engage not only with our researchers, but also to engage with a lot of the institutions and companies, biopharma, biotech companies that are around New York. And of course, nowadays, with remote capabilities, we can collaborate with many people around the world, but having the in-person capabilities to really discuss and engage in collaborations is very important, and it really helps to move the technology and the technique flow cytometry forward. Very briefly, our lab is composed by an amazing group of people. I've been fortunate to find a lot of great people, that some that have come and gone, and follow their opportunities. But still, it's a rich environment where we have also we're lucky enough to have so many people go through our lab and give us so many great ideas and contributions, not only to the lab, to the core facility, but also to the research being done at MSK, which is very important and really makes me very proud. We have several instruments, 16 instruments. We're moving towards mostly spectral flow cytometry. So most of the instruments will be spectral instruments in the near future. But we serve about 250 labs, and all these metrics don't seem very impressive. I'm sure that all of you have at least one of the metrics that's way superior than ours. You probably have more instruments, probably have more staff, probably have more users. But altogether, we do have a lot of usage. So here's just an example. We do about 400 sorts a month, which actually gives us a lot of opportunities to engage with our researchers and come up with really interesting projects in collaboration, but also ideas for us to improve and optimize the technology and the technique. I know that all of you must have, in your labs, or at least in the core facilities, a mission statement. I thought I'd put the mission statement of our lab here. I think it's very important to, especially in this context of what I call R&D, research and development. Most of the research, I would say, all of the research that we're involved in is through collaborations with other labs. We don't do our own research, but we do a lot of development. We do a lot of technical projects that can be just small improvements to actually pushing the envelope of the technology. And I wanted to highlight the fact that, a lot of cores focus on centralizing instrumentation and providing the support on the instrumentation, but it's very important to note that we also need to provide high-level technical and scientific expertise. Of course, technical expertise, I think most of you already provide by maintaining the instrument performing well, etc. But what I mean with scientific expertise is understanding at least enough of the biology and the scientific questions that that are important to bridge that knowledge gap between the scientists and the technologists. And so we try to do that as much as possible. I'll give you an example. If you're building a panel for someone, you need to understand a little bit of the biology behind it, right? What are the markers that you're going to use? What are the most important ones for, to answer that scientific question? So, for us to really, and this is what I'm highlighting here, to advance the scope of MSK research, or, to advance the scope of any institutional research, it's very important to have this in mind, right? That you want to push the boundaries of the science that's being done in your institution by pushing the boundaries of the technology. Now, a lot of people have asked me, why do you involve yourself in R&D projects? Just for curiosity? Absolutely. I mean, that's an important aspect. Most of us are scientists, and we continue to be scientists and asking questions. And if we see that things are not working well, we tend to try to figure out why they're not working. But for me, in the context of a core facility, R&D projects are very important. If you think of your staff, or if you think of a team, it's very important for continuous learning, for them to be able to, for us to be able to continuously learn the science, the technology, be abreast of the latest advancements, but also to acquire and improve lab skills. Some of us started in full cytometry without any research experience, and it's very hard to give that experience because we're mostly operating instruments, etc. Right? So giving the opportunity to actually improve those lab techniques are important. Also, to create some empathy towards the researchers. I mean, this is something that happens many times. I remember there was a staff, from my previous lab that was always asking, why don't they just start sorting in the morning? Why doesn't anybody sort really in the morning? Why do they only start at 11 or 12? Well, because it takes a long time to prepare samples to sort, and depending on the tissue that you're trying to isolate, etc. But it's important to also understand the pressure that scientists are. Why don't they bring always controls? And it's interesting that when my team started working in projects, some of the first questions they asked was, do I really need these controls? Because that's a lot of work. And this really gives us a notion of what's the importance of a control balanced with, how much work does it take, and how necessary it is, right? So it really gives a different perspective when you are actually teaching and providing training to researchers. Also, for staff recognition, this is obvious, right? You have a chance to go to meetings, present your work, you contribute to, you feel that you're making a very small contribution, but you feel you're contributing to science in general, right, and to research. And obviously, lastly, it increases collaborations in cutting edge science and technology. And this is obviously good for the science, it's good for the lab, because again, it enables them to learn and acquire those skills that are very important in general. Here are just a few examples of projects that we are currently working or worked in the past. Since I started at MSK, we've been working on a universal QC platform. We've made huge advantages. The last person working on this was Ben, who left for Thermo Fisher, but we are continuing to follow this goal. Another project that we're working on is to build an autofluorescence signature repository on different spectral instruments. The idea is to then start working on, let's see the impact of using fixatives or different, processing methods, etc. We also started building a repository of successful sample prep protocols to share with everyone. This came from the idea that we keep reinventing the wheel. Every time somebody wants to sort neurons, it's almost like we have to restart and do research and figure out the best conditions. And especially in a big lab, it's kind of difficult to share all that knowledge to everyone every time one person is involved in a project. The other thing is we've been working on beads versus cells. I won't go into details because Barbara will present that, but we've been trying to see in what conditions do beads work better than cells. We've worked also on a project we called Well Tracker. This is essentially an idea that came from Simon Monard, and Mark built this little contraption that he'll also show to be able to monitor drifts and shifts of the ACDU and the BDFX area, which can actually be implemented in any of these plate deposition units. Thomas Miller, when he was with in our core, worked on Easy Tisney. I thought that was a really nice project. The idea was to help people to optimize multidimensional data analysis, finding shortcuts and just making it easier for people to, breaking that energy barrier that's required to really understand multidimensional data analysis. We have an online sort time and and this was volume calculator, to calculate the amount of volume that your tube can contain. And I'll quickly go to social media platform. This is something that was very important for us to be able to enhance the outreach of all the educational content that we were putting out, but also to attract and retain talent. Magda will talk about that. We started working on some videos on SOPs for instrument troubleshooting. Mark will talk about his 3D printing projects, which has really branched. He's been doing a lot in terms of 3D printing, and I'm sure he can help you guys a lot in doing that and implementing that in your labs. We've implemented a panel design service. I'm not just talking about providing panel designs to people, but actually building a pipeline that we can, at every stage, we can, for instance, have somebody, we can delegate certain steps to certain people, set timelines, review, even have different people in the lab come up with different options and see which one is best. We haven't done that, of course, because we're short on resources. I wish, but that's an idea, right? If you're able to do something like that, and et cetera, et cetera. And actually, I forgot. I started taking medication for my pain, and I know that I moved this up to put more projects, but eventually I forgot about the rest. But we do have many other small or bigger projects going on in the facility, and this is just the team that's been working on them, and all the names that I could remember very quickly that have been involved in these projects that I just mentioned. A lot of people that we've, like I said, some of them were in the lab that while they were in the lab, they were working on these projects, but also a lot of external collaborators, which is really great. We share our knowledge, but we also bring a lot of knowledge to the lab, and we grow with that. And so with that, I think I've taken already too much time, but I'd like to introduce Magda. I don't see her coming to the stage, but I'll just mention that Magda was hired obviously as a flow cytometry technician, technologist, resource scientist, however you want to call But one of the challenges I gave to Magda was to build a social media platform, like I said, to be able to share the education the content that we generate. It's a huge waste not to provide that content to, and this actually started with Kathy Daniels and her work on the Flow Post-its. This was one of the motivations to put also the social media platform, and also, as she'll explain, to attract and retain talent. The bigger the social presence is of your core, the easier it is to attract people because they know what you do or what you're doing, and even to retain, because you can share the stuff that people are doing in the lab. Okay, so I don't know, Magda, are you ready?
Thank you, Rui, for the introduction. My name is Magdalena, and we'll just switch to.
Stop sharing.
I see. Okay, I need to stop sharing. My mouse is not it's not working. I'm trying. I swear. There we go. Okay. And we have some time for questions at the end.
Okay, perfect. It's a pleasure to be here today. My name is Magdalena, and my side project in the lab, it's managing social media platforms, just like Rui introduced. So we'll talk today a little bit about this. Okay, to realize the importance of social media, I would like to start with the numbers, because there is almost 5 billion people and social media users worldwide, which means that that's 59% of the global population. And on top of that, people are spending nearly 2.5 hours daily in front of their screen. So these numbers are already powerful. When we were starting over 3 years ago, our initial objectives was to have access to all the latest news and informations, to know what events are happening. Also, we wanted to use this platform as a source of networking and a place where we can network, and then it eventually can lead to collaborations, and also overall the community engagement. On top of that, we also had a lot of educational materials that we wanted to share, and we thought that, okay, this is going to be a great place when we can do that. And even on top of that, we already had very well established websites, so we wanted to drive more traffic towards that, so people can use and have all these informations. As we also learned, and as a part of the strategy was like, okay, can we use social media as a recruitment tool? And the answer is yes, and I will mention a little bit about it later. So now the question is like, okay, how to start your own social media presence without like a full digital marketing team, right? The first part is the strategy. So when we were at first thinking, we had to find the inspiration. So we were looking at the lab, other course, other websites. And then during the process, we also wanted to find something unique that can help to distinguish us from others. That's what Flow Post-it is for us, and I hope some of you might have heard about it. Then, when we were also choosing the platforms, there was already so many available platforms out there, so starting with all of them would be absolutely overwhelming. So we chose the ones that were the most familiar to us, and only once we had a well-functioning pipeline, we started adding new ones. From the strategy, once it's well thought, we can move on to the practical parts. So first thing, we need to set up the profile. So we need to put as many information as possible, fill in all the fields, all the keywords, all the bio, make sure that we are linking our website that we have, or maybe cross-linking other social media platforms, have a clear and short description of who we are, what we do. the location. Also very important is to make sure that we have consistent logo across all the platforms. So when we start with one, it's good to think already ahead and have something universal. And also very important part is to also take into account the right images sizes of all the background photos, etc. Then the second step would be to follow colleagues and leaders in the field. And when we've done that, we start to realize like, okay, we know the yearly schedule, we know what events are happening, and also we kind of are on top of the newest informations, the latest technology, the latest technologies. So we felt like, okay, this is actually super helpful. Then the third step, it's very important, is to post content frequently. It's either can be scheduled ahead or live, like today. It's a great opportunity to share some information from our meeting, and also at the appropriate time. And now, what is the appropriate time and why is it so important? Well, I took a data from Sprout Socials, and you can see the global engagement across different platforms. And as you can see, the highest engagements are the darkest spot. The highest engagement for Instagram doesn't necessarily mean that this is the highest engagement for TikTok, right? And why is it so important? Well, you can have a wonderful content that you want to share, but if you post it at 3 a.m. in the morning, it will just get lost in the social online space. So this is definitely a good starting point, and then from there on, we can take and test what works for us. Hashtags and tagging other accounts. Hashtags or keywords are helping to find the relevant content back, and then tagging people and other accounts, it also makes it broader. You can reach more audience. Sharing relevant content consistently. So back to Flow Posted series. And to give you a background, we started posting in January 2020, every month. In October 2021, you can take a look in this blue box that we have a tweet impressions, which means like how many times it showed on people's feed. Those tweets were on monthly around 6,000. It got 6,000 impressions. In April 2022, these numbers absolutely increased and got to almost 25,000 impressions, which is very nice and very rewarding. There are a lot of time-saving strategies, a lot of management tools, which I mentioned here. But what I would like to stress and highlight here is to use the team effort, because this is absolutely not one person job. My whole team contributes tremendously. They are spending a lot of time working on it. They are learning from that. We all learning from working on those. So if you have that opportunity, definitely take it. Also, another very important part is to make sure, and we also take it very seriously, before we post anything, all the informations are going through really strict revision, because at the end of the day, it's our also responsibility to make sure that the content is valid, is correct, and we don't put any misleading informations. So now let's move on to the results. Like, how do I know it actually works, right? So that's what scientists likes the most, which is analysis. Each platform has its own analytics where you can take a look and see. And this is super important to after a while go there, check and take improvement, because maybe coming back to the tweet impressions, maybe you can learn that, okay, maybe the text alone doesn't really work. Maybe I should add the picture, maybe the picture also is not enough. Maybe I should create a little video to attract more views. The second one is the national reputation and recognition. And we absolutely realized that it helped to establish our global recognition. Our team members are associated with social media, are also engaged on many occasions. And also when we took a look at the data from Google Analytics, it actually told us that our website has attracted visitors from 70 countries, which is very cool. Equal education and scientific engagement. That is something that was very important for us from the very beginning. And it indeed helped to spread the content, especially the Flow Posted series. those where we shared, people are coming back with questions to us. They are also increasing the scientific engagement, and also it helps to initiate collaborations. And then lastly, which is very interesting, since we started our social media presence, we absolutely noticed a trend in our recruitment process. So during the time that we are present, among 10 new hires, 9, they applied because they learned about us through social media. And that was a part of the strategy. And I'm pretty sure that there's a lot of people in this room who would love their recruitment process to be a little bit faster. And additionally, on top of the fact that with every new position, we have way more CVs than we had to the times when we didn't have the social media. These are not just CVs. These are also very relevant CVs that are coming in. So with all that, I would love to acknowledge the whole team who contributes a lot and continuously, and also Rui, who gave me this opportunity to work on this project. And I would like to introduce Mark, who works on a very cool stuff, and his project involves the 3D printer. Thank you.
Hello, my name is Mark, and I would like to talk to you about developing 3D printing solutions and developments for flow cytometry. There we go. So why do 3D printing? What is the purpose of it? I'm pretty confident to say in saying that we have all walked into our lab and found some issue or problem or something else nagging that you wish you could change. And following that, we have always done what we've done best, and that's find a workaround to it. Now, with the advent of 3D printing and the current state it is in, we can actually do something about those nagging problems and actually have customized solutions to all of them. Whether it's for storage, organization, instrument improvements, specific solutions for users, or tools to make things life a bit simpler, 3D printing has a lot of things it can offer, as you can see here on this poster that was presented at Cyto. I will go into further detail what you're seeing here. So what does it take to do some 3D printing? Well, the workflow is pretty simple. First, you pretty much come up with an idea. You walk into the lab and you see something that is not to your liking. This idea then gets drawn into 3D design software, and afterwards gets sent to another piece of software that translates it to instruction for the 3D printer. After a while, you will finally get your printed product and your solution. Now, skipping the part of the idea because everybody has those, we move on to the 3D design software. There's a lot of them around, as you can see, and this is only a small selection of those. Over at Sloan Kettering, we settled for FreeCAD. Now, why did we do that? It has a lot of things going on for it. Next to that, the name already implies that it is free. It also has many design features that have that are more towards, more professional softwares. It also is open source, which allows for features to be downloaded from a large database that is produced by a very dedicated group of users. That also things like if you need screw tabs for a thing, you can just download the add-on. It integrates automatically and you're done with it. Now, the one drawback to this software is that, as you can see, it is a little bit more complicated and has a bit of a steep learning curve. A lot of icons and menus are thrown at you, and I can probably already see a few people thinking, yeah, am I going to be able to even learn this? Well, to be honest, we already are working with very complex software as it is, so why not this one? Everybody can do it if you work with any of our flow cytometry software. Um, th- uh, to be honest, this is the biggest hurdle to overcome, and you don't have to start out with a very complex design. My first design personally was actually just a block with 3 holes and a couple of numbers printed on it. So start simple to begin with. Now, the other must-have, and I will emphasize on this, is yes, a set of calipers is an absolute must. You want precision measuring if you want to get into 3D printing. You'll thank me later. Now, reasons for the hardware we are using is currently the Ultimaker S3. A few Christmas ago that we were told we could get a 3D printer and it had to stay under the capital equipment budget, and we made very sure to use all of that. So this is a little bit of a higher end product than most, but don't think that any other 3D printer that is out there is going to be less capable of doing the work for you. Now, some of the reasons for this printer were the dual extrusion, which allows for multiple, actually 2 different types of material or 2 different colors of materials to be printed at the same time. Also, that includes things like PVA, which is water soluble and allows for more complex designs to be printed. The next thing also is, of course, the swappable print cores, which allow for different materials. So you're not just stuck to PLA, which is a very hard plastic, but also softer plastics, as well as that PVA I was talking about. Next to that, of course, a large print bed is very convenient when you want to print something very large or just do mass production of certain items that you just happen to need. And Cura, the software that comes with it, is the software that sends the instructions to the 3D printer, and it's highly rated and absolutely a pleasure to work with. Having said all this, I would like to go to a few examples. As you can see here, there are examples for storage solution or organizing and decluttering. Custom tube racks specific for instruments with all of the holders for nozzles, tube holders, etc. Also, you can see a custom bead box, which makes it for our users very easy to find the right beads they need to use for that specific instrument. And on the bottom right, you see the little block with 3 numbers and 3 holes. The next is solutions to sorting needs. Currently, we made a custom collection tube holder for BDFax Aria with 2 5mm tubes and 2 15mm tubes, just for those particular sort that have a large bulk and some smaller population that a user wants as well. Custom PCR plates, because all brands are a little bit different and not all of them fit correctly. So you need a custom solution for all of them. Also, the Sony SH800 was not capable of sorting into a Terasaki well plate, and that has now a holder as well, so we are able to sort into that. Instrument improvements. So custom panels and brackets for valve systems for various instruments, making it very easy to start up and shut down any instrument. This one is for the Aurora SCS, but there's also for the SH800, and on the bottom is the BD-S6. These brackets also work very well for our BD Arias. Also, camera holders for observing collection tubes and sample tubes that can be then monitored in software that is very easy to install and very convenient to keep an eye on anything. The latest project is not completely finished yet, but is a customized replacement door for the Sony ID7000. The door swung out a little bit too much to our liking, so I am replacing it with a sliding door. Instrument improvements, and I would like to put well tracker on, and hopefully this video will work. Does that work? Yes. So well tracker is actually a tool to monitor it's a 3D printed bracket with a camera and a laser that monitors your plate sorting. This one is particularly good for BDS6 and the Fax Symphony and the BD Fax Aria. These are the current designs for the holders, and it's good for monitoring any form of drift, skipped wells, skipped rows, and that sort of thing. I would like to acknowledge Simon Menard for the original idea for that. Tools. Some simple tools here are the interlock bypasses for the BD Fax Aria to let laser light through while you have the hood open, as well as the interlock bypass for the Sony ID7000. The more complicated tools are this one right here, which is a scope to look at laser flow cell laser intercept points, and is currently fitting not only Arias, but also for Tessas, Fax Symphony S6, Aurora, and Aurora CS, and is an absolute must if you want to align your lasers, or if you happen to have a laser that needs replacement. I would like to conclude this presentation with that you can go to our website, fccf.mskcc.org, and find more out about our project and a link to our 3D design repository on Thingiverse. All of the designs that you have seen here are freely available there for download, along with a lot more. So feel free to print them for yourself, but I hope that this presentation has inspired you to maybe try some designing yourself and get into 3D printing. Now I would like to introduce my colleague Barbara, and of course acknowledge all of my colleagues at MSK. But over to Barbara with assessing beads as universal single stain controls.
Hello. How does this work? Is it like this? No. No, the pointer. Who said arrows? Yeah, I'm trying. Perfect. Thank you. So it's a pleasure to present my project about assessing beads as universal single stain controls. And I'd like to start by going to when we're setting up a multicolor flow cytometry experiment, we have so many steps since the biological question, all the different steps that we have for panel design, sample prepping, staining, and then acquiring, analyzing our data, and finally publishing it. Even if that all is a lot of steps that we have to think of with a lot of mini steps inside, I want to just focus on the staining part. So during the staining, we already have to worry about antibody validation, antibody titration, doing our FMO controls, optimizing our buffer in case of polymer dyes, and then optimizing our single stain controls. So the idea of having a universal particle here was not only to decrease the time we spend optimizing the single stain controls, but also to increase our data quality and the reproducibility between all of the multicolor flow cytometry experiments. So to have single stain controls is crucial to do our compensation or our mixing. So ideally we use cells, the same cell type with the same antibodies, conjugated with the same fluorochromes from the same vial in case of the tandem dyes, and just use that as your single stain controls. But due to very different reasons, sometimes that's not possible, even because of the marker expression is too low, unknown, or it's simple variables between samples. We also have on the other side of this spectrum that we don't have enough cells sometimes because the samples are too precious, or we don't need, or we cannot grow enough to have for the samples and for also for the single color controls. So another part of the motivation was also to have a particle that could use in these cases. So the method that we developed to achieve or to try to find this universal particle was by staining some cells and beads separately with anti-human CD4 with different fluorochromes and running them on the Cytoflex and or the Aurora after the vendor QC passed. We got two different matrices. One that used the beads as a single stain and another one that we use the cells as a single stain that we applied to the concatenated single stain cells. So then with these concatenated files, we evaluated if the unmixing and the compensation was calculated accurately. And the way we did it was with a collaboration, we developed this method or this metric that we call secondary stain index, that if you see here the formula, it's based on the stain index formula, but instead of getting the values from the primary channel to see the resolution, we're going to see the values from the secondary channel here and evaluate if the compensation or the mixing was calculated accurately. So if you look again to the formula, we see that it's divided by 2 times the RSD, meaning that if the value is between -1 and 1, it's within these 2 SDs, and we consider it to be accurately unmixed or compensated. If it's outside this range, meaning that it's above 1 or below -1, we don't consider that it's accurately unmixed or calculated. Another way to see or to evaluate these particles was by calculating the spectral correlation coefficient, which is equivalent to the similarity index. And we normalized all of the spectra for all of the fluors and overlay them when beads and cells were the carriers and compare. So previously, we presented this poster at Cyto 2019, where we evaluated the difference between cells and beads as compensation and unmixing controls. We used lyophilized PBMCs and UltraComp Plus beads, and we tested 22 different fluorochromes that are all listed here. The first thing we look at was the spectral correlation coefficient, which again is equivalent to the similarity index. And we noticed that some of the fluors are below 1, meaning that when the cell or the bead is carrying this fluor, the spectra changes. When it's equal to 1, the changes are none. When we look to the secondary staining index, that's a metric that I showed before, we noticed that for cells, we don't have any unmixing or compensation inaccuracies. We just have inaccuracies when using beads, and they are lighted here in yellow. But I want you to look specifically to this PSI5 example. While we see some unmixing inaccuracies here, depending on the secondary that we use, but when you go back and look at the spectral correlation coefficient, it's equal to 1. So this, it's not enough to detect potential unmixing inaccuracies. So most recently, we continued this project and we added the SpectraComp as one of the particles that we tested. And we started by testing these 6, because from the previous data, were already the ones that were giving more unmixing inaccuracies. So the first thing we noticed when starting this project with these 3 carrier types was that the SpectraComp negative peak was shifting compared to the universal and stain when we added the antibody. So the first thing we thought was, well, maybe this will impact how the unmixing accuracy is going to be calculated. So when we applied both matrix, one that we draw this gate from the negative and another one that we just use the universal and stain, we didn't see any differences. So using the negative gates or the universal and stain does not impact the results. When we look at the spectral correlation coefficient, we see that for the SpectraComp, they are all very close to 1 for all the fluorochromes that we tested. while the UltraComp Plus have some variation compared to the SpectroComp. And then looking at the brightness of the different particles, both beads, the UltraComp and the SpectroComp, are brighter than the cells that we use, being the SpectroComp the brightest among all. This is a visualization of our data for all of the 6 colors that we tested. And each graph represents each fluor as a primary against all the secondary fluors that we test as represented on the x-axis. And on the y-axis, we have the secondary stain index that I explained before, that it's between -1 and 1, and that's why these dotted lines, it means that it's accurately unmixed. So just to give a little bit more visualization on this data, this is how the data looks like if the secondary stain index is below -1, meaning that it's not accurately unmixed. When it's within -1 and 1, and we consider that it's accurately unmixed, and when it's above +1, and we also don't consider that it's accurately unmixed. So going back now to all the data that we have, this visualization help, we can see that for all of them, the cells are always working, and they are accurately unmixing all of our combinations. While when we use beads, we have some inaccuracies depending on the combination that we use. And then I would like to highlight that sometimes it will do an excellent job of accurately unmixing the samples. And then sometimes it will also not work. So this comes to the conclusion that the difference in the spectral correlation coefficient can highlight potential problems, but cannot tell about all of them. So using an additional metric like the secondary stain index will allow to better evaluate these unmixed or these compensation inaccuracies. And the impacts on the different fluors and different particles can vary from day to day and require multiple testing to build confidence in the particle-fluor combination to each experiment. Then we would only consider using the synthetic particles as single stain controls when the cells are not available for the reasons that I mentioned in the beginning. I would like to thank all my team that is here today, especially Rui for letting me presenting my data today, and to Kathy Daniels that started all of this amazing project, with Joanna, that both are alumni from our core facility, and José Mario Morgado, who developed the secondary stain index metric. I'd like to give some resources. My email is here if you are interested in continuing this project, collaborating with us. You can download all of the slides from us on this QR code if you want to stay tuned from our project in our website, and both posters that we just published or we presented at Cyto. So we're happy if there's time to take some questions.
Great work. You guys are up to some really exciting stuff. Any questions on Zoom? Who do you want to go to first? Okay, in here. Great. Sorry, thank you.
Hi there. So based on what you have just described, are you saying that you are consistently using cells all the time when you can?
That's a great question. No. First of all, we work at the core facility, so we test this for you, people that really want to use the beads. We try to have a comparison of cells and beads and see what works better at this point, because we cannot find something that is universal and can be straightforward tested. I'm sorry, but I cannot hear you.
How many times did you wash the beads?
That's a great question. 2 times with the cells as well. I treated the cells the same way I treated the beads as well.
Hey, Barbara. Hey, so just following up on this, right? And I know what you found. So it's really interesting work in how you've quantified this, but where do you think you'll take it from here? Do you think that all of these errors are going to be significant to the actual gating of a sample? Or have you maybe started looking into that or thought about that for next steps? Because as you said, right, even without having a great universal control, there are going to be times when people need beads or, basically. So if you have these errors, and not saying that they're not there, how would you best recommend working around them? And did either of the particles seem to give any trends to you on whether they'd work better with certain floors or the others?
It's also a great question. The goal here was to have a universal particle, so we don't have to test for every time that we have to build a new panel. So meaning that when we don't have a choice and we have to use beads, we have to test them, and we have to test the different floors. I'm not saying that, for example, for this panel, which is already the worst case scenario, because we know that are the ones that give the most problems when we're trying to do the unmixing or the compensation. It would be good to have one particle for this. Not having that, we'll just have to test what we have in the market. So the future work here would be to wait for the companies to develop or a new particle or to improve the ones that are in the market so far.
So two questions, one serious, one not at all. First, does the unmixing math matter? Like, did the Aurora versus another spectral instrument make a difference? And second, Mark, can you just 3D print some better beads for us? That'd be much better.
Give me time.
That's also a great question. We were looking forward to tests across all of the spectral instruments that we have, but the idea here was to have a universal particle. So we started in one, which was the Aurora, and it didn't work so far. So we'll wait for the beads and then test the other instruments.
Can you guys hear me? Can you hear me?
Yes, we can hear you, Rui.
Okay. Yeah, I was just gonna add something to that. So when we looked at unmixing, we also looked at conventional or polychromatic instruments like the Cytoflex, and we saw the same issues with compensation. So it doesn't seem to be an issue necessarily with the unmixing algorithms. We did test initially when Kathy was working on that. We were testing also with FlowJo, and the errors that we were spotting were the same. I mean, we didn't see any difference there in terms of unmixing inaccuracies.
Great job. Thank you guys so much. There we go.